mutation. This is a mutation that we found that causes early onset Alzheimer's disease. It is very rare, like all of the early onset mutations, but it causes Alzheimer's disease with 100% certainty. So what I'm showing you here, you don't need to know the details of DNA sequencing. You can just look at the pattern on the left, which is a healthy individual, and the pattern on the right, which is his brother with Alzheimer's disease. And if you just compare the two patterns, you can see that there's a difference right here. This individual has just 2 bands. This individual has 4 at this point. Can everybody see that clearly enough? What we're looking at here is the fact that we're looking at chromosome 21, a specific part of the chromosome 21 DNA sequence. It's actually a part of the amyloid gene sequence. And at this particular point on that DNA sequence, in the individual with Alzheimer's disease, one of his chromosomes has the normal sequence, but one has the abnormal sequence. This is just 2 spelling mistakes out of about 3.5 billion letters in the genetic code. And this causes Alzheimer's disease with 100% certainty. Now, the significance of finding mutations like this is that it really focused us on amyloid as a central cause to Alzheimer's disease. When we introduced this mutation into cells in the laboratory, what we discovered was that this mutation causes amyloid to be produced at about 6 or 8 times the amount it normally is. So you imagine if you've had this mutation from birth, that's why you get early onset Alzheimer's disease, because you've been building up extra amounts of amyloid all that time. Whereas in the more common form of Alzheimer's disease, folks start to build up amyloid in the brain once our systems start to wind down, once those mechanisms that pull amyloid out of the brain start to wind down, that's when it starts to build up. One of the key things that happened when the mutations were identified was that we were able to now model Alzheimer's disease in animals. And so we used transgenic approaches, which allow you to introduce human genetic material into a mouse. And so what we have is a mouse that has his entire normal genetic code, and then he also has a piece of human DNA as well. And that mouse has the Swedish mutation, that human sequence of the amyloid gene. And those mice are born normally and they develop normally. But as they get older, they start to develop amyloid plaques in the brain. This on the right is the transgenic mouse. This is a human Alzheimer patient. And the stain that we're using here to detect the amyloid plaque is COX-2. It's an inflammatory marker. So you can see that when we look at inflammation around the amyloid plaque in the human and in the mouse, it's very, very similar. We see other things with these mice too. They don't get all of the features of human Alzheimer's disease, but they do exhibit memory problems and learning problems as they get older. So once you have an animal model, that means you can now start to really test your theories about what's causing the disease and how you can stop it. So developing these transgenic mice really revolutionized the Alzheimer research field. And here's an example. There he is. A very reproducible experiment. Put a male mouse and a female mouse together, and you end up with a few thousand in a very short space of time. And you'll see our animal facilities as you walk around today. This is a mouse performing one of those memory tasks I mentioned. This is the so-called Morris water maze, and I'll mention that now because I'm going to come back to it later. What we do is we have a kid-sized paddling pool, and it's filled with water, and there's a platform below the surface of the water. And mice don't like to swim, and so when you put them in the water, they'll swim around, and when they find the platform, they'll climb out. And so we have markers like this on the side of the pool. And healthy mice will learn over time that the platform is next to that marker. So when you put them into the pool, they will swim straight there and they'll climb out on the platform. But the mice that have Alzheimer's disease will not learn. They may still find the platform by chance, but there will be no evident learning, and they won't remember one day to the next that the platform was next to that marker. So, a number of ways to approach the